So it's a pleasure to see so many people here today. So welcome to the David James seminar. So Dr. David James, he was an administrator in the department and did great service to the department. He also left a legacy to the department, which is doing fantastic work supporting various studentships and also enables us to get fantastic speakers in. And so, yeah, this is the top seminar in the annual calendar of Department of Pharmacology. We run ones almost every week in term, so please look out for those. If you're not from pharmacology, and very pleased to see you here. So we've got quite a few people online, so welcome to them. And, we'll be, if you can enter your questions in the chat, we'll be very happy to ask some of those at the end. And following the seminar, we're going to have drinks and Italian food in the cafe at the top floor of the department. So also hope people come and join us there. And so, yeah, I'm delighted to welcome Feng Zhang. Dr. Zhang is a molecular biologist focused on improving human health and very famous for the key role in development of the two really revolutionary technologies of optogenetics and CRISPR-Cas systems. And so this included pioneering the use of Cas9 for genome editing and discovering CRISPR-Cas12 and Cas13 systems, and then taking them forwards for therapeutic and diagnostic applications. And so we're very much looking forward to hearing about exploration of biological development.
Thank you. Thank you so much. Well, thank you so much for the invitation to be here. It's really wonderful to be here today and to interact with everyone. I've met so many great researchers and students, and the discussion has been really thoroughly enjoyable and rewarding. So I'm from the other Cambridge, and it's nice to also be here at the original Cambridge. And so I prepared a couple of things from what we have been working on, and hopefully we'll have a nice discussion and maybe even foster some future collaborations together. So the overarching theme of my research, somehow this is not working. Okay, so the overarching theme of my group's research is developing genetic medicine. And so genetic medicine is a very powerful concept. So I first got inspired by this when I was in high school. So I worked in a gene therapy lab when I was a high school student. And there, working with the scientists and researchers there, I learned about this idea that you can now rationally, using the new principles in molecular biology, to be able to design therapeutics based on your understanding of the fundamental mechanism of disease, and to be able to cure diseases rather than just masking the disease symptom. So I usually put up this picture of a rocket delivering a satellite into outer space to kind of illustrate this idea of modularity. Genetic medicine, very much like this situation here, is a two-component system. There is a vehicle like the rocket, and then there's a payload, which can be a variety of different molecules that you deliver into the cell to be able to modulate it or modify it to achieve the therapeutic outcome. So vehicles include viral vectors or lipid nanoparticles or any other sort of gene delivery methodology. And then the different payloads can span from siRNA to antisense oligos to mRNA to gene editors to epigenetic modifiers and many other ways to be able to modulate cell state and cell function. So the holy grail is to then have a toolbox, a variety of different vehicles that can go through all the different cell types specifically, and also be able to target to different tissues. And you can mix and match those vehicles with different payloads to be able to achieve the desired therapeutic outcome. And so that's kind of the goal that we have been working on, to really sort of build as much of this toolbox as possible. So the recent advances in gene editing and also in payload development has far outpaced the advances in vehicle technology. And so, in fact, nowadays, the biggest bottleneck for genetic medicine is the lack of targeted and also efficient delivery systems for many different tissues. And so that's what we have been sort of spending a lot of time thinking about and working on. And so today I wanted to tell you about two areas that we have been working on that hopefully provide new approaches to develop gene delivery methodologies. So the first story centers around proteins that are found in our own genome that are very ancient, that were acquired from previous retroviral infections, and then we can understand and harness them to turn them into gene delivery vehicles. And the second story is analogous. It's about viral-like things that bacteria have acquired long time ago through evolution. And then we can harness these and engineer them to be able to deliver protein payloads into cells for therapeutic uses. So let me tell you about kind of the first story. So this is kind of the current slate of delivery technologies for genetics. So there are viral-based systems, and also there are non-viral-based systems. So they each have pros and cons. The viral-based systems are very efficient, and they can be targeted to a few different tissues. But the biggest bottleneck is that sometimes they're immunogenic, and also sometimes they have a lot of difficulty to be able to scale up. So it's hard to make enough of this to be able to really dose sort of entire sort of large animals, including human patients. The non-delivery system, the non-viral delivery system are also very powerful, but it also has drawbacks. So it's powerful because it's very scalable. So the COVID vaccine, which is a form of mRNA LNP therapeutic, has been really scaled up. Billions of people receive this mRNA medicine, and so it's very powerful. But it also has limitations in the sense that it's mostly targeting dendritic cells in the immune system or targeting hepatic cells in the liver. And that is because as a lipid-based system, through ApoE mediated uptake, it largely goes into hepatocytes, and it's hard to get it to go to other tissues. So it would be nice if we could develop a system that can integrate both of these systems' advantages, right? So something that is targetable and efficient, but then at the same time scalable, like the lipid nanoparticle. And so we turn to these human proteins because we think that they may have the ability to realize this sort of synthesis of the benefits of the two systems. So these proteins were first discovered when the human genome was first sequenced. So when the human genome was completed, scientists, especially bioinformaticians, began to analyze all the different protein coding genes. And so this study here by Campilas and colleagues, they looked at some of the 20,000 protein coding genes from the human genome and looked to see which ones of these have strong similarity to viral proteins, especially the structural protein from the retrovirus GAG. So this is what they found. They found different families of genes in the human proteome that have very strong structural and sequence homology to the retroviral structural protein GAG. And so these are just some of these. And this one particular protein, ARC, which is expressed in the brain, is thought to be important for memory and learning and also synaptic plasticity, has been demonstrated a few years ago by Jason Shepard's group and also Travis Thompson's group, that this protein found in the brain, it forms virus-like structures. And so Jason's group studied the mouse version and Travis's group studied the Drosophila version. And in Drosophila, they showed that it can package mRNA, and it can also traffic this mRNA across the neuromuscular junction. It's kind of a very interesting and exciting discovery. So I saw these two studies and I started to think, okay, maybe this broad family of proteins all have similar behaviors. Maybe they do different things, and we can harness them to be able to package nucleic acids and then deliver them to downstream cells for different applications. So Mikey, who is a postdoc, he has his own lab at Harvard now, and also Blake, who is a senior graduate student in the lab, they teamed up to begin to study this family of proteins. They first chose a few of these different human GAG proteins, and they found that biochemically they form capsids. And these are cryo-EM images, and you can see that for some of these proteins, recombinant versions of the protein can self-assemble into virus-like structures. And so these are what these capsids look like under EM. So that is very exciting because it shows that not just ARC, but many of these other proteins in human, they all form virus-like structures. So then they try to see whether or not they form virus-like structures that are released by cells. And so what they found is that they tried a number of different human genes, and they found that a few different ones, when you heterologously express them in HEK cells, which is human 23-cell line, many of them secrete virus-like particles into the supernatant. And so that's good. And that means that at least some of these in this sort of unnatural setup is able to release virus-like structure, just like what ARC has been reported to do. And in 293 cells, this protein called PEG10 is most robustly secreted. And so it's always easy to study things that have a strong signal. So we just focus on PEG10 for our downstream work. And we package these PEG10 particles in the 293 cell lines, harvested them from the supernatant, and then we extracted RNA to see, do they package specific mRNA? And if they do, what is this RNA? It turned out that the most abundant RNA that's packaged is the mRNA for PEG10, for the gene itself. So this, at first, was kind of confusing, but if you think about it, it makes a lot of sense. Because these PEG10 genes, they originated from viruses, and they package the viral genome. And so the PEG10 mRNA is really the evolved version of that virus's genome. And so seeing this specificity, selectivity kind of makes sense. So then by studying this more, we then kind of figured out that there are specific motifs on the RNA that gets packaged. So that allowed us to then develop a system where we can use HEK cells to package things and to be able to deliver this sort of the mRNA that we're trying to deliver into different cells. And so the way that works is very similar to how you would package a lentiviral vector system. We introduce into 293 cells a vector that expresses the PEG10 protein, and then also a membrane fusogen. So this is what allows the particle to bind to a recipient cell and then enter into the recipient cell. And then for the third vector, we encode something that will express the mRNA that we're trying to package. So we figured out that the 5' UTR and also the 3' UTR are signals on the mRNA that allows us to get packaged. And so we then express our gene of interest flanked by these packaging signals so that it will get loaded into the viral particle. So when you do that, transfect everything into HEK cells, you can produce these virus-like particles, and they will get secreted into supernatant. So then you can harvest these, and they are almost lentiviral-like things, except they package mRNA and they deliver mRNA, and are done. And these are all produced using human endogenous proteins. If you put this under the electron microscope, you will see that the particles that are released look just like any retroviral-like particle, and they're different from the exosomes that are secreted by these cells. So the negative control image, these are exosomes, and these are the PEG10 particles. We did sort of gold nanoparticle staining to make sure that these are indeed the PEG10 particles. So by putting these particles onto cells, you can mediate delivery of therapeutic relevant mRNA. And so here we deliver the guide RNA for Cas9 along with the mRNA for Cas9. So we'll make the protein and we'll deliver the guide RNA. And so when you do that, you can get a reasonable level of the gene editing in the recipient cells. So this shows that this is a viable delivery system. So we got very excited once this happened, and then we started to think about how do we scale this up so that we can test it in animal models and see if we can use it to really treat diseases in an animal model context. When we started to do that, we then realized that this system suffers from the same scalability challenge that other cellularly produced viral particles suffer from, which is that it's hard to make a lot of it. And so just to give you a sense of scale, this experiment was done in a 96-well plate. And so you have individual wells on a 96-well plate. Each well has about 5,000 to 10,000 cells, somewhere in that range. To transduce those cells in a single well, you need to have an entire 15-centimeter plate of supernatant, and you concentrate it down into 20 microliters. You put out the cells, and you get 40% or so gene editing. So my students were giving my meeting to update on the research progress. We're talking about how to make this and test it in a mouse. So they showed a slide live meeting. It was very memorable. It was a picture looking at Earth from the outer space, and there was a stack of petri dishes showing the height. It turned out that if you do a back of the envelope calculation, if you want to produce enough particles to treat a cohort of say 5 experimental mice and 5 control mice, you have to have a stack of 15-centimeter plates that's as tall as what will exceed the geosynchronous orbit. And so that clearly that wasn't a viable approach to scale to an animal experiment. So then we went back to the drawing board and we thought, how do we make this system work better? And so this is something that Blake has continued to work on. So rather than using cells to produce it, we thought, okay, if these particles are able to assemble if these proteins are able to assemble into particles recombinantly in vitro, maybe we can develop an in vitro system where we can use bacterial production to make tons and tons of the protein. And then we can just in vitro spontaneously assemble particles just like lipid nanoparticles are assembled. So this is kind of the approach. So we use in vitro production to make the mRNA, and this is a scalable process because for COVID vaccine production, you can use in vitro transcription to make lots and lots of the spike protein mRNA. So we can make mRNA in the same way. And then at the same time, we can use E. coli. We can make many grams of the protein just by doing E. coli monoculture. And so this is also a scalable process. It's also not very expensive. And if you mix the in vitro transcribed mRNA along with recombinantly purified protein, these will self-assemble into virus-like structures. And this allows us to produce it at a much larger scale, orders of magnitude more than we are able to using cells. So that's kind of the basic idea, is that we can then make these sort of synthetic particles. So we first tested to see how this idea would work. We began by using the Drosophila R protein as one of the more easily purified and expressed proteins. And so this protein behaves very well. So in the absence of mRNA, the protein doesn't self-assemble. So you don't get particles formed. And if you just spike in some mRNA into that protein solution, you start to see the protein particles getting formed. And so this is very nice. So this is actually an advantage over some of the viral systems, because things like AAV, they form empty capsids. And then a helicase loads the DNA genome into that AAV capsid. But what happens is that when you produce AAV, you end up with sometimes half of the particles as empty particles. And that also means that if you're delivering AAV into a patient, you could be delivering a lot of sort of not very useful viral protein into the patient, instead of just delivering as much as possible the therapeutic. So anyway, so this means that we can make sure that almost 100% of the particles have the payload loaded, and so we're not sort of being inefficient about delivering sort of unnecessary protein. And this packaging is done by charge-based interaction. So RNA, mRNA is negatively charged, and then the inside surface of this capsid is positively charged, so that the two charges come together and they can form capsids. So taking that idea further, we can also package the Cas9 RNA ribonucleotide protein complex. And so here is Cas9 protein only. You don't see packaging. If you just load the guide RNA, so just have the negatively charged guide RNA, you get packaging. But then if you provide the Cas9 protein guide RNA RNP, these can also get packaged. So this means that there is enough negative charge on the Cas9 guide RNA RNP to still allow for packaging. So this is a sort of a nice function. With LNP, you cannot package RNPs. You can package RNA, but you cannot package the protein along with the RNA. So this is another difference between this system and the RNP-based system. So we put these particles onto BJ fibroblast cells just to see whether or not the Cas9 RNP is functional. And so here again, you can get. For doing this experiment, we didn't have to grow lots and lots of cells to then transduce the cells. This is just a tiny bit of what you can produce using this in vitro assembly process. So because the Drosophila ARC protein has been shown in Drosophila to mediate delivery across the neuromuscular junction, we thought maybe let's inject this into muscle in mice and see whether or not they get taken up by muscle cells. And so in this case, what we did is that we tried to turn on a reporter protein. And so the reporter is tandem dimer tomato. So over here on top, we're using the Drosophila ARC virus-like particles to deliver the Cre mRNA. And the Cre would get expressed and then turn on the M-cherry reporter or tandem dimer tomato reporter. And then for comparison, we also injected a lipid nanoparticle delivering the Cre mRNA as well. So what we find is that for both the DARC and also LNP, we can get activation of the reporter expression, which means that we can functionally deliver mRNA into the cells. But the toxicity levels are very different. So with the DARC particles, you can see that the muscle fibers are intact and they look pretty healthy. And then you get expression of reporter in these muscle fibers. But if you use LNP, there's quite a bit of toxicity. So the muscle fibers, they don't look very healthy. And you can see these are really not very sort of healthy looking muscle cells anymore with LNP delivery. So that's something that was very interesting to observe. And then we thought, okay, if we can get into muscle cells, maybe we can try to see if we can correct the splicing mutation in a muscular dystrophy mouse model. And so here we took the DMD mouse model and then we injected either the Cas9 RNP or Cas9 mRNA plus guide RNA. So these are just two different ways of introducing the gene editing machinery. And here we provided two guide RNAs or two RNPs, because what we're trying to do is we're trying to use two guide RNAs to cut out this splicing mutation. So what we find is that if you inject PBS, you don't get restored expression of dystrophin. But then with either RNP or with mRNA delivery, you get restoration of the dystrophin expression. So this shows that we can use the system to then restore expression of dystrophin in these muscle cells. And we quantified it a little bit, and we found that the RNP approach is more efficient than the mRNA approach. This is still early data, but it just shows that if you can deliver already made RNP, it's able to work more efficiently. The mRNA system is slower because mRNA has to get expressed. And while the mRNA is getting expressed, some of the guide RNA could get degraded in the cell. so you're kind of less efficient. And so this is probably a little better. So then we thought, okay, in addition to getting to muscle cells, we also want to get into other cells. So maybe we can work on trying to target it to different cells. And so here we use the spike catcher system to be able to functionalize the surface of our sort of dark particles with antibodies. And so here, what we found is that you can pretty efficiently put about 6 molecules of the antibody per capsid. And you don't need too many more. So 6 molecules, even fewer than that, is actually enough to confer targeting specificity. So to test how well this works, we try to target T cells in mice in vivo. And so here in the legend, we have either dark with something that doesn't target T cells, just IgG, or we have anti-CD3 antibody that is able to target CD4 and also CD8. And then for comparison, we also have LNP, non-targeting, also a targeting LNP. So what we found is this. So this is a reporter activation in our reporter mouse to see if we're getting targeting T cells. So if you look at the dark conditions, we're able to get good targeting in both CD4 and also CD8 T cells, kind of in the same order of magnitude as the LNP targeting. But then if you look at non-T cells, the CD11b positive population, you can see LNP gets there, but then our dark particle doesn't get it. So you can get good targeting, better targeting, more specific targeting than the LNP-based system. This is in the blood cells. We know that LNP is getting to the liver very robustly, so we also look at the liver. So this is sections of the liver from the same mice. You can see with the tandem dimer tomato reporter activation, the LNP basically caused the entire liver to become red. And the dark particle didn't target the liver cells. And so this also showed that you can get good targeting in the T cells, and you can avoid targeting hepatocytes. So this provides a better way to achieve cell type specific expression than a lipid nanoparticle-based approach. So that's what we're continuing to work on, and I think it's kind of a promising approach to use these in vitro assembled protein nanoparticles to be able to deliver therapeutic payloads into cells. Okay, so let me shift gear a little bit and tell you about the second story, and this is largely the work of Joe Christ, who is another senior graduate student in the lab, and he's been working on these sort of prokaryotic injection systems. So these systems are called Photorhabdis virulence cassettes. So Photorhabdis is a type of bacteria. It's a pathogenic bacteria in insects. And this is the cryo-EM structure from this one of these systems solved by Zhang and colleagues. And you can see that molecularly they look like a big syringe. And they have a hollow core on the inside, and you can load proteins into this hollow space cavity, and that's what gets loaded into cells. And this is a schematic kind of illustrating how this system works. In the fully loaded condition, you have this long syringe-like structure. It's got a sharp spike, and it's got these gray tail fibers that provide recognition of the target cell. Upon sort of bombarding onto a target cell, the tail fibers get unfolded, and they will recognize the target cell surface. And then this recognition engages this spring-loaded mechanism to then compress. When it compresses, the spike protein gets shoved through the cell membrane, and then the payload that's in this shaft also gets unloaded into the target cell. So this is how this system works. So we wanted to see if we can harness the system. This system naturally works on insect cells. So we thought maybe we can take it and engineer it so that they can work on human cells. So the first thing we did is that we took all of the genes for making the system from Photorhabdus bacteria and transfer it into E. coli. The E. coli is much easier to work with. And so by doing some tweaks, we figure out that we can express the whole system. And these are EM pictures of the syringes that E. coli is able to make. So they look very much like what Photorhabdus produces, just different long syringes secreted by these bacteria. So we tested to see whether or not these E. coli-produced bacteria, these syringes do work. And so these are different conditions, and this is the experimental condition where we have the wild type syringe, so unmodified, and it loads a toxin that would kill insect cells. That's what the natural syringes do. And you can see in these SF9 insect cells, most of the cells got killed by the toxin. And then if you carry out a few controls, like you delete the payload, so no toxin. If you delete the payload loader, so the toxin doesn't get loaded into it, or if you get rid of tail fibers, none of these kill the insect cells. So this just shows that we have a system that we can produce using E. coli, and it injects the toxin or the protein payload into the inside recipient cells. So next we thought, okay, maybe we can reprogram it to recognize human cells instead of insect cells. The tail fiber, this structure here, is what mediates recognition of the target cell. And so we thought, okay, we need to change this so that we can convert it from an insect cell recognition domain to a human cell recognition domain. And so the way we do that is that we modify the wild type tail fiber, and we try to modify this domain. So this is AlphaFold 2 generated structure. So we started this project before AlphaFold 2 came out. And Joe probably tried more than 100 different designs, and they all failed because we didn't have a structure which is randomly guessed at where we can make changes. So before we were ready to give up, AlphaFold 2 came out. So then we put the sequence into AlphaFold 2, and then we got a structure. And so we designed one more attempt. And so we thought, okay, we can make a cut right here to get rid of the binding domain, the natural binding domain, and we can fuse different new synthetic binding domains onto this tail fiber. And this eventually worked. So we either fused an Ad5, adenovirus 5 binding domain to this tail fiber, or a EGFR DARPin onto this tail fiber to target EGFR on human cell surface. So this is the experiment. So we now have a human reporter cell line. This is A549. This is a lung cancer cell line. It's targetable by Ad5, and it's also targetable, and also expresses EGFR on surface. So the wild type tail fiber, also the payload is now pre-recombinant rather than the toxin. So the wild type tail fiber didn't result in any reporter activation, and if we get rid of the binding domain, there's also no reporter activation. And then once you put Ad5 binding domain on it, you can get GFP to turn up. And also the EGFR DARPin turn GFP on. So this told us that we can use this approach to then retarget the system to be able to deliver proteins into human cells. We can also load Cas9 or zinc finger deaminase into the syringes or inject something that's more useful to the companies. I should mention that in this case, we are only able to load the Cas9 protein. So the guide RNA had to be separately introduced into those cells in order to get edited. We also tested how well the system would deliver across different targeting domains. And so we generated different syringes with different tags inserted onto the tail fiber. So these are different tags: HA tag, flag tag, EE tag, etc. We also made reporter cell lines that express antibodies on the cell surface. And so we have different antibodies to recognize the HA, flag, etc. And so we found that you can get very good specificity. So you only really get reporter activation when the tag that's on the tail fiber on the syringe matches the antibody that's on the reporter cell surface. So this tells us that we can get pretty good specificity for this type of delivery. We also inject it into the mouse brain. So here we use the AT5 binding domain. We put it into the hippocampus, and you can get some targeting of cells in the hippocampus, and so on. So this still needs much more work because the biodistribution of this injection is very limited. I think part of it is because we're working with large sort of syringes that don't diffuse very easily, and so we're trying to figure out better ways to do that. And so what I'll show you for some of our continuing work might be this thing. So so far the approach that we have taken is to load the protein into the shaft, and then the syringe will then push the protein to the cell. We can load RNA, we can load DNA, so we wanted to figure out a way to diversify the payload that we can load. So the strategy that we came up with is inspired by a different system. This is the type 6 secretion system, which is a different system. This is usually a membrane-bound injection system. So for the type 6 secretion system, the payload is actually covalently linked to the tip of the injection apparatus. And so when this tip gets punctured through the cell membrane, the payload gets dragged in because the payloads are linked to it. And so we thought, okay, maybe we can modify our system. We can use the tip, the spike of our system to deliver a payload. And so the spike structure is encoded by two different proteins, PVC8 and also PVC10. So we thought if we fuse our payload protein onto these spike proteins, then we can use the spike to drag it into the cell. So we devised these different strategies. We put GFP onto PVC10. This allows you to load one molecule. Or you can fuse onto both termini, and you can get two molecules. And then if you fuse it onto PVC8, because there are multiple PVC8 monomers in this spike, you can get about 4 molecules onto the spike. So this is what we have been thinking about to diversify things. So this also means that we can use chemical conjugation or use spike catcher, spike tag to then load other things onto here, such as oligos or even RNPs. So that's kind of the idea. So we tried this out. So we thought, this is how we're going to do it. We'll fuse it onto the tip and then just use the tip to drag it into the cell. The tip falls off of the syringe once it's inside the cell. This is how the payload then diffuse to the right place in the cell to carry out the function. And so we tried this. We fused Cas9 onto PVC10 or PVC8, and you can see on EM, this is a wild type syringe, and this is with one molecule Cas9 RNP tethered to the site, and you can get it fused onto the C terminus or instrument, or you can see you can get 2 molecules of this RNP onto the tip. You can also get 4 molecules onto this PVC8 fusion. So these are RNPs. So they are Cas9 protein bound to the guide RNAs. It's the fully active complex. And so we tested this on reporter cells, and we can inject it into cells and get some detectable levels of gene editing. So we still have to optimize it more, but it shows that we can use this approach to get fully functional Cas9 RNP particles into the cell. So anyway, so that's some of the work that we're working on. We're continuing to mine from matrix diversity because both these human GAG proteins as well as the syringes, they really all came from these natural diversity. And so we are continuing to mine this microbial diversity to find other systems that potentially help us solve and advance this therapeutic delivery need. So this is about sort of half of the direction in my lab. And then recently we have started to do a bit more work in a new area. So I'll tell you a story that we've been working on in this new area. And that is about engineering resilience. As people get older, resilience in our tissues, in our cells, start to decline. And sort of getting older or age is also probably one of the most strongest factors that influence disease risk. So as you get older, all these problems come up. And so we thought maybe we can figure out ways to improve resilience through engineering of cells in our body. So the work that one of these things that we have been trying is to make the immune system more resilient. And so we all know that the immune system is very important. When we're first born, the immune system doesn't really have any memory. And so it takes some time to develop the immune memory. And so in the early days of immune development, our immune system gets exposed to different things, viruses, bacteria. We get sick a lot, or babies get sick a lot. Parents also get sick a lot because the baby brings viruses home. And so that's when the immune system is developing. And then once the immune system gets developed, it enters into this long period of homeostasis. It's able to help our body maintain health by fighting off infections and clearing sort of bad things from our body. But then eventually the immune system starts to decline. And so in old age, homeostatic functions decline, and then we start to lose the ability to have all these protection. This is when inflammation goes up, when cancer risk goes up, and also when our ability to fight off infections goes down. So this is kind of the natural course of how immune systems develop and decline. So we thought if we can prolong this stage of homeostasis, then we can probably also prolong health. We can reduce cancer risk in old age. We can make our body fight off infections more efficiently also. So it turns out that one of the organs that's really important for immune homeostasis or immune development is the thymus. And so the thymus is this organ that is very well developed in young age, but it starts to degenerate very early on. In fact, it's one of the first organs in the body to begin to degenerate. So this is a section of the thymus from a 17-year-old, so it's just a little bit after puberty. And you can see that the thymal tissue has already degenerated quite a bit. And certainly by old age, even by someone who's 35 or so, the thymus is mostly not functioning anymore. It's mostly degenerated. So by 70 years old, the thymus is all gone. So the thymus is very important for developing new T cells. And so as the thymus goes away, our ability to develop new T cells to replenish the diversity of TCRs diminishes, and that contributes to our inability to kind of have strong surveillance and protection. So we thought maybe we can try to reconstitute thymus function. It was hard to reconstitute the thymus or to regenerate the thymus. And so what Miracle did is that he thought, okay, maybe we can work together to get other organs to provide more function, more thymic function. And so the idea then was to use the liver to express genes that are normally expressed in the thymus, so that even in old age, we can use the liver to provide some of the function that the degenerative thymus used to provide. And so by profiling young and middle age and old thymus, we found a number of different genes that are diminishing as the thymus degenerated. And so eventually we settled on 3 different factors. And so we used mRNA and a lipid nanoparticles to package them up. And then this is able to deliver those 3 factors into the liver to then express them. The liver is an interesting place for 2 reasons. Number one, our entire blood supply flows through the liver. They also flow through the thymus. And so if there are factors that the thymus is producing, when those cells flow through the liver, they get exposed to those factors as well. And second, during embryonic development, the liver actually plays an important role in developing the immune system. And so in adult, we thought maybe we can try to reawaken some of these functions. And so the 3 mRNA that we settled on are DLL1, FLT3L, and also IL-7. So DLL1 and IL-7 are secreted, and FLT3L is a surface ligand. And so we first, tested to make sure that we can get good expression from this mRNA approach, and indeed we're able to get a decent mRNA expression. So then we dosed sort of late middle-aged mice, about 1-year-old mice with these 3 factors. And then we use a vaccination challenge to see whether or not we're able to improve the immune response in these sort of thymic factor treated mice. And so this is what we did. So white is just vaccination in wild type mice. And then red is vaccination in mice that received a controlled luciferase mRNA, so without expressing the thymic factors. And then blue is vaccination in mice treated with the thymic factors. So in a wild type mouse, if you vaccinate across a range of different ages, you get different vaccination response. And in fact, they follow a very nice, almost linear distribution. So when they're young, you get more antigen reactive T cells. And then when they're older, you get far fewer. So basically, if you take any mouse, if you didn't know how old the mouse is, you vaccinate it with this peptide antigen, and you measure the T cell response, you can plot the response along this curve, and you can estimate the biological age of that mouse. So that's how this works. And so we then compared the mice, 1-year-old mice that were treated with either the thymic factors or with the luciferase control mRNA. And we found that if you treat the mouse with this thymic factor, the vaccination response is significantly boosted. In fact, is more comparable to a mouse that's about half of this age. And then if you vaccinate the luciferase treated mice, it's the same age or same response as mouse with its own biological age. This shows that you can rejuvenate thymic function. So then we tested to see whether or not, if we're boosting T cell function, are we also able to boost immunotherapy response in a cancer model? And so we took mice, and then we treated the mice with these thymic factors over the course of 3 weeks just to boost the thymic function. And then we implanted tumor cells into these animals. And then we gave the animals immunotherapy, so basically anti-PD-L1. And then we thought to see, do we affect the efficacy of this? And so what we found is that with the thymic factor treatment, we can reduce the tumor volume in these mice, which suggests that the T cells are able to work better, or the immunotherapy is working better in this animal. And then also, we're able to extend the lifespan. So whereas if you treat with the luciferase control mRNA, all the mice eventually die after about 2.5 weeks. But then if you treat with thymic factors, almost 40% of the mice are able to continue to survive. And so this sort of shows that we can also make the immune system work better in conjunction with immunotherapy. So anyway, so that's still an ongoing work. But mainly we're just very excited about the ability to develop systems from naturally occurring molecular mechanisms, turn them into tools. And also looking at biological systems and identifying ways to change cell state and function, and combining it with genetic therapy to be able to improve sort of biological resilience and improve human health. Last but not least, this is my team at MIT. We're supported by a number of different groups, and then really excited to be here, and really great to have an opportunity to tell you about our work. Thank you again.
Thank you so much, really inspiring. So, got some questions.
A really nice talk. I'm curious with the capsid proteins, how important is it for them to be human? And do you have any idea of the immunogenicity in humans?
Yeah, that's a really great question. So we don't know the full answer yet, but the motivation is that because there are human proteins, they would already be sort of tolerated by our immune system, and then we can then do redosing without worrying about repeated immunogenicity. So we still have to do more testing there. But so far in mice, it looks promising. Even with the Drosophila arc protein in mice, it doesn't seem to be all that immunogenic. Although I think I have to do more studies. Sure.
Another question?
Earlier in the workflow of your question about the early workflow of your labs, genetic diversity to find
Yeah.
new programmable systems.
Uh-huh.
Are you guys using, and you mentioned
Uh-huh.
a recent project, are you guys using some of these models to improve that pipeline? So identifying sequences with homology to known programmable systems and then
Yeah.
that selection by predicting the folding of those sequences.
Yeah, absolutely. So that's a good question. With all the exciting advances in AI and language-based models, we do use that in our mining. I'm giving another lecture at 6 at Trinity. So I'm talking about some of those work this evening. So using structure-based prediction, you can zero in on a very small but critical domain for protein. And you can take that domain, for example, the RNA binding domain Cas9, and you can mine the entire structure space for any protein that carries any resemblance of that domain. And when you do that, you can find a vast universe of proteins, and some of those have similar RNA guided activity. And so, based on that kind of approach, you can then discover a lot of very interesting systems. So I'll talk about one of them. We'll call it Tiger-Cas, which is a brand new RNA guided system.
Are you guys open sourcing some of these pipelines and workflows, bioinformatic workflows?
Yeah, we upload we share the code with some of these, but they're complicated to set up. It's not easy. You have to do some serious software development to turn it into a package that someone can just download and install on a computer.
Plug and play. Yeah.
That's right.
Okay, we'll take a question from online. So Chinku's work has asked, for the thymic factor system, has it shown to increase thymus size and the T cell production? Are these effects driven by T cells or by boosted B cells as well?
Yeah, that's a great question. So we look at the thymus. Expressing these thymic factors in the liver doesn't increase the size of the thymus. So I think we're just really just driving T cell effects through the factors that are made by NK cells. And then and then in terms of whether or not it's T cell or B cell mediated function, I think we're seeing sort of T cell mediated function. Although we have to do more experiment to look at what is changing in T cell. We see changes in the T cell receptor repertoire in these mice. We don't see so much the B cell changes in these mice. So that's why I'm basing the statement that we're seeing mostly a T cell effect, but we have to do more work for sure.
To follow up from that, so
Yeah.
the centenarians have larger thymuses?
That I don't know. That's a good question. Not sure. Yeah. Yeah, I don't know. That would be very interesting to look at.
That's it.
Are you saying that you're wanting to sort of make the liver act a bit like how it was in the embryo? Is there any possible concerns that it may then trigger cancer?
Yeah, that's a great question, and that's certainly a valid concern. In our mice, we haven't seen any issue there. I think it's worth noting that the expression of these thymic factors is very transient. So each LNP injection probably only lasts for 24 hours. And we do about 7 injections over the course of 3 weeks. It's brief pulses of these factors. If you are constitutively expressing them in liver for a long time, I think that might be a different scenario. But I think it's important to limit the exposure.
I think that ARC protein is important for like learning. So when you use them as like the basis for this people, for a potential gene therapy, have you also noticed like any effect of the vehicle on like genetic related processes?
Yeah, that's a great question. So we haven't tested that, but I don't think you will see a learning memory impact through the way that we're using it. The mechanisms through which ARC mediates synaptic plasticity or memory consolidation is not known. And what we're using these for is to deliver a completely different payload. Normally they deliver ARC mRNA, so we're not delivering ARC mRNA. And so that's why I don't think they are going to give us the same effect of an actual ARC function. But we'll have to study more.
And can they cross the blood-brain barrier at all?
Not these particles. Yeah, we haven't seen them. But I think there are ways to engineer them as the BBB. Either using antibody-mediated engagement of transferrin receptor, or we can evolve them in the same way that AAVs have evolved to change the avidity and get it across BBB.
I was wondering with the ARC system, are you delivering linearized RNA, or can you deliver circ or self-replicating RNA as part of the?
Yeah, great question. So in this example, we have only delivered linear mRNA. I think you should be able to also deliver circular RNA. We just haven't tried that. We haven't explored self-replicating RNA because they are potentially immunogenic, RDRP, and double-stranded RNA intermediate, and that can be potentially problematic.
Thank you.
Hi, great talk. Thank you very much. And then broad range of topics. For the type 6, if I remember correctly, where you showed that you can basically fuse molecules to the spike. I was curious, how did you measure, or do you see an effect of what the nature or the size of the molecule that you're attaching to the spike is, and how that affects the efficiency of the injection? So in other words, will that be a universal system in your mind that you can attach whatever you want, or will there some constraints that will kind of limit the use of the system?
Yeah, that's an important question. We haven't looked at it systematically yet. So we have tried GFP, we have tried Cas9. Cas9 is about 4 times, 5 times the size of GFP. So it is a meaningfully larger, and both work. I'm sure there's a limit. You can't use it to inject things that are too big. So yes, but where that sort of sharp cutoff is, I don't.
Because you also mentioned that you were thinking about DNA, RNA. So here I'm also thinking about the charge that will kind of play a role, right? So
Yeah. So we have tested antisense oligo delivery for short DNA oligo as a homologous recombination template, and that works. Yeah. And you can use it for siRNA delivery.
Questions? Yeah, regarding the famous factor, have you think about looking at the like a long-term memory effect of what they will cause in the immune system, or like the metabolic change of the system, and also about the aging? Are they going to like transient exposure for like 7 times? Are they going to make the mice live longer?
Yeah, so we're doing those studies now. We don't know yet. The challenge with aging experiments is that it takes a long time to run. But we have noticed something interesting. So for the mice that are treated with these factors, we see hair regeneration. Also, we see repigmentation of fur. So like, gray hair mice become dark again. So it's kind of interesting. And those are, it's unclear what the mechanism is. We're also trying to figure that out. But it may not be a T-cell mediated mechanism. It could be a different mechanism.
Any more questions?
Speaking about the prokaryotic system to inject, when you comment on mining genomes from new prokaryotes, do you think that are systems that naturally evolve with CRISPR systems to inject systems? And if you are mining any relationship of any naturally evolving injection CRISPR or just injection systems in general?
Yeah, that's a great question. I don't know, but it's entirely possible because CRISPR is CRISPR. People say that it's a bacterial immune system, but you also find CRISPR in viruses. So phages also use CRISPR to target hosts to either provide host suppression or to suppress competition with other phages that also infect the same host. So there is a lot of function that have been being explored. I suppose computationally you could look for coexistence in these systems and maybe to look for Cas9s or CRISPR systems that may have acquire signal peptides that would allow them to get loaded into cells. That's very interesting. I don't know. Yeah. And maybe even if you don't find it with this type of system, with the PVCs, you might find it with other secretion systems. Because bacteria have many types of secretion systems.